Thank you for participating in our second quarter 2025 business and financial report. Here I am making forward-looking statement. So just briefly about the Modalis for those who are new to Modalis. We are the first epigenetic company using a CRISPR and developing therapeutic gene therapy for a genetic disorder. And the technology we called CRISPR Gundam has been validated in multiple species, and we have demonstrated efficacy without serious toxicity. And based on that technology, we have developed a pipeline primarily for muscular dystrophies. On top of that, we developed a product for CNS disorder and also cardiovascular because the technology has unlimited potential for other area. And also, importantly, we have established manufacturing process and for the challenging new capsid and enable tissue-tropic delivery for lead programs. And that is supported by experienced team based in Massachusetts, United States. And also our technology is covered by strong IP portfolio. So this is showing what the epigenetic editing is, but essentially that is the transcriptional control of the target gene. So for a genetic disorder caused by excessive amount of gene expression, we turn off the gene. And for those genetic disorder caused by less amount of or a lack of the expression of the target gene, we express the gene so that we can compensate the missing function. So unlike the gene editing technology, which cleaves the DNA, and also there are also other gene therapy modalities, but the epigenome editing is diversified from the other tools. And we using a virus vector to deliver our molecule to target tissue. This is important because we want to see the efficacy solely in the target tissue, and we don't want to touch on the other tissues. So it's really important to use the tissue-tropic delivery tool. And also we have a multi-layer mechanism to limit efficacy and also safety concern other than the target tissues. So by that, we are able to establish single dose therapy, which efficacy lasts long, and it's not just the symptom care, but it's the disease modifying effect. So that's the advantage of CRISPR Gundam. So among the epigenome editing company, we are pretty much differentiated from the others because we are not just the first company, but also we focus on upregulation, whereas the other company's primary target is suppression of the gene. And we think that the advantage of the epigenome editing is more clear in upregulation because for suppression, there are a lot of other modalities that can do so. So I think uniqueness of the technology is more vivid in gene activation rather than suppression. So here we are presenting the 5 things as listed here. First, we're going to start with the topics of second quarter 2025, followed by the financial update and the fundraising we announced yesterday. And also that comes followed by the growth strategy, and lastly, summary, and then Q&A. So let me begin with a key point of second quarter 2025. We have a lot to tell you in this quarter. First of all, we have, as we announced at the end of June, we have a revised timeline, which I'm going to tell you later. And the second point is the access to new technology, which we are able to avoid liver toxicity caused by the virus vector. And thirdly, we are presenting the positive annual proof of concept data in our DMD program, MDO-201. And also we got a grant from a two group for our FSHD program. And then lastly, finance and others. The first, this is the current pipeline. Nothing changed from the last quarter. I'm gonna skip this and then jump into the READ program update. So the READ program is again for Rama2 congenital muscular dystrophy, which is a devastating disorder. Most of the patients do not survive past adolescence. So it's a really severe disorder. And this disorder is caused by a mutation in a gene called RAMA2. And so if the patient has a mutation in the gene or lack the whole gene, it leads to a severe muscular dystrophy. And the first point, as I mentioned earlier, we have announced the timeline revision at the end of June. This is due to multiple causes, but mostly logistical problem. The reason, first reason is that after we got the funding last year, we restart the whole development process, but that caused a lag for restarting the program. That's the first reason. And the second reason is that we changed a major contractor for one of our work. So that makes the certain impact on our timeline too. And then lastly, there is some delay in the procurement of materials. So including all those three things and also other minor things, this caused the timeline delay. And we reschedule our IND target to sometime in 2025. But I wanted to, emphasize that this is not due to a technical issue. So it's all about the logistical issue. So we can overcome the issues, and also we are able to file IND 2025. The detailed timeline is still under review, so we're gonna share the timing when in 2026 will be announced later after we fix it. And for manufacturing, we have a progress here, and we already announced that we have established the manufacturing process. But after that, we have gone through the scale-up process, and we were able to manufacture the material at the scale we're gonna make the clinical material and also the following commercialized material too. And as a result, we were able to gain the good productivity, quality, and yield. So we are really happy with that. So that process and will be used for our GMP manufacturing. So here I wanted to take a moment to talk about the capsid license. So as people are aware that there is growing attention to liver toxicity to the AAV capsid, which is when it used in high dose systemic injection, right? The most of the problem is due to the liver toxicity of the capsid. There is a huge demand to avoid that issue, and we are able to achieve that, get the access to the new technology, which called MyoAAV, developed by Broad Institute, so that we can avoid that issue. So this is the engineered capsid. So unlike the natural occurring capsid, which are captured mostly at liver. The new engineered capsid is tropic to muscle tissue, you know, 2 digits more than the natural occurring capsid. So that means that we can deliver more capsid to target tissue, which is muscle, rather than delivering it to liver. So that has a huge benefit for efficacy side and also safety side as well. And we have been negotiating with Broad Institute for a long time, but we have reached the conclusion, and then we are able to get the license of the MyoAB capsid for development of our MDL-101 program. And we have concluded the license term, which is not undisclosable, but I can tell that Medallic is going to pay a royalty and a milestone as our development progress. So this is a huge opportunity, not just for us, but for the patient, because we are able, with this state-of-the-art technology, we're able to avoid the toxicity issue, which people have a lot of attention to. And then next page is about the trial design, which hasn't been changed since we presented this last time. But just briefly, this is a two-dose step study. We started with raw dose, and then after we completed the second dose and also follow-up period, we're gonna discuss it with the external group so that if we can go up to higher dose for the entire rest of the patient or the part of the patient. So then there's three ongoing natural history study. So we compare with those results, we'll be able to compare our safety and also efficacy of the molecule in the patient. So that's the overall, the high-level study design. Okay, so including that, MDL-101 made a huge progress in the last quarter. We have completed tech transfer to CDMO and scaled that up to 200 liter, which is the size of the manufacturing for GMP. And we have completed pilot manufacturing and got incredible parameter, including the productivity and the yield as well as the quality. The progress smoothly toward the GMP manufacturing. So we are really happy with the manufacturing side. And also on top of that, we have a progress on the IND enabling studies. As we presented earlier, we have a two species study ongoing. One is a mice IND enabling study that has been initiated, and also we have an NHPDL tox study, which is also initiated. And also, we have ongoing coordination with the patient group as well as the agency, so to prepare for the coming clinical trial. So our current strategy is to go to United States. That's still our primary strategy. However, we are seeing a lot more other opportunity in the non-US country. So we are communicating with the patient group as well as the agency in those countries so that we try to see the feasibility of running the trial instead or concurrent to United States. Okay, shifting gear, and I wanted to talk about our program for Duchenne muscular dystrophy or a DMD disorder. This disorder is another type of muscular dystrophy, but the prevalence of which is much higher than RUM2 CMD, which is 1 in 3,500 to 5,000. So it's a very more popular muscular dystrophy. And this disorder is caused by a mutation in a gene called dystrophin, which is very located in the intracellular portion of the muscle cell. So if the patient has mutation in it, in the dystrophin gene, which function as the shock absorber of muscle, that leads to the dystrophy of the patient, just like LAMP2 CMD. But the disease-causing gene or protein are the different. So there are a bunch of the attempt to develop the therapeutic for DMD, including the one which already got the regulatory approval in United States. But those approach are based on the concept of packing mini dystrophin gene instead of the wild type dystrophin gene, because the dystrophin gene is a very big size gene with the size of 11 kilobase pair. So it can't fit into AAV vector, which has the packing capacity of 4.5 kilobase pair. So it's about 3 times more than the packing capacity. But there was a finding that there is a milder type of symptom in certain patient, and the scientists got found that those patient has mini version of dystrophin gene. So then there are several groups try to develop therapeutic with the mini dystrophin genes listed here. They are all different kind of mini dystrophin gene, but it lacks the big component inside of the dystrophin gene. So it has a limited function compared with the whole size, big size dystrophin gene. But our approach are so unique because instead of putting in the dystrophin gene, we activate utrophin gene, which is the featured version of dystrophin gene. As you can see in the bottom of the slide, the utrophin gene is pretty much, very much highly homologous to dystrophin gene, and it includes the component intermediate in the intermediate region. So it has been proved that utrophin gene compensate certain function of dystrophin gene. But still, it's a big size gene. So what we can do with our technology. So this is how it works. So in the patient, in the newborn, the utrophin gene is actually active, whereas the dystrophin gene is off. But after the birth, the switch is crossover, and then utrophin gene shuts down, whereas the dystrophin gene kicks in. But if the patient has mutation in the dystrophin gene, that leads to dysfunction. But what we can do with our Gundam technology is to reactivate the utrophin gene to restore the dysfunction of dystrophin gene. Since utrophin gene has the most of the domain very common to the dystrophin gene, our hope is to restore the problem in the DMD patient. And here we are able to prove that's true in the disease model mice. So we are using disease model mice called MDX mice and test our molecule with the benchmark molecule. So most to the left, that's the wild type, which is shown as BL10 beagle. This is the wild type mouse, which is completely function normally in the treadmill, as shown in the upper right. So we dose for the mice group at the age of 4 week old, we dose the molecule or we dose the beagle and see how it functions after 5 week period in the treadmill analysis. As you can see here, the disease model mice, which is shown as a MDX vehicle, did not have enough walking distance in this analysis. But when the mice are treated to the most right, as shown in green, it has recovered to the certain level. So this is the benchmark molecule as like a Elevidys, which has a microdystrophin packed in RH74 and dosed at 1.33 E 14 VG per kg. So just like the clinical dose. So it has recovered, right? But we have tested our surrogate model molecule of 201 for mice version. And as you can see here, we are able to establish the recovery with much lower dose. As you can see here, even at 2E13 per VG per kg, we are able to see the recovery in the mice um, nearly to the level that the surrogate microdystrophin molecule perform. So this make us convinced with the data. So we are trying to get more readout in the ongoing study and the following study too. So we are very much excited to see this result. Even before this study, we have a strong confidence that we would able to see the result because in the previous version of the capsule with AB9, we also see the efficacy, and also the same system is used for MD-101, our read program. So that system is combined with the new myotropic capsule, we had a strong confidence, but we actually saw the incredible result here in this disease model. Okay, so then next disorder is FSHD. This is another type of muscular dystrophy. This is a relatively milder symptom, but patients are suffering from the lack of the muscle function. So I don't go into the detail of the disorder, but this disorder is caused by the mutation in DAX4 gene, which is toxic gene in muscle. But this DAX4 gene is coded in chromosome 4 and expressed in the very early stage of the development, but it shut down as the baby grow. But due to some reason, if the regulation of off regulation is released, then the toxic gene product uh, has is expressed in the muscle tissue, and it damaged to the muscle cell that leads to the problem in the patient. But what we can do is our Gundam technology is to shutting down this expression of the DAX4 gene, and this is a very you know realistic approach and also powerful approach to treat this genetic disorder. Okay, so we have got significant data for this FSHD program, but we have been suffering from the lack of finance to develop this molecule. But the program has been awarded uh, uh, from two grants. One is X Prize, which is the prize for not just for a healthcare problem, but also for other problems like environment problem and also development, space development. So we were able to get the grant money for our FSHD program from the X Prize, and we are one of the 8 groups that have got the grant money to FSHD. On top of that, we got also funding from the grant group called Solve FSHD, which is established by the founder of Lululemon, Chip Wilson, and he put in his own money for the development of the solution to FSHD. So we have already got the molecule with the selected read guide RNA, and we have filed a patent. And we have, with this money, we have restarted our MDO-103 program with a drug candidate for the treatment. And we have corroborated with the academic group to run the disease model of FSHD. So I think we'll be ready to share the data in a year or so. Okay, so I think this is the last slide for the research part. So with those progress, we have done the presentation just recently at the Scientific Family Conference called SciFi 2025, and we have shared the data, including the one for MDA-101 and with the new mesotropic capsid. And also, we have a plan to present at the sixth generation genome editing therapeutic summit in October. And then this is the revised coming milestones. As I mentioned earlier, we have revised our IND target to sometime in 2026 for MDO-101 program. And also, we will be announce the coming milestone for the DMD and FSD program once we get a clearer idea on the development timeline. So including that, this is also the summary of the achievements so far for the programs and also coming milestone. So on top of that data we share today, we expect the additional readout for MDO-201. So we're gonna share that certain time and possibly in the Genome Editing Summit in October. We also made the great progress in the IP side, and our RAMA2 CMD patent, which had been granted in Japan, has been granted in United States. Was granted in United States in July. So this is the proof that our technology and also product has their uniqueness and also inventive step in terms of the patent viewpoint. All right, then next part is financial report and also explanation of the finance we announced yesterday. And this is the balance sheet of our finance in the Japanese yen. So I'm gonna put that in the US dollars so that the people can understand it a little bit easily. So we have a current cash position of 1.7 million US dollar in a bank account, and which is a little bit lower than the one at the end of 2024. But we have enough cash to cover our development activity in the coming 12 months. This is the PL again in Japanese yen, so I converted it in the US dollar. So the point is that we have spent 6.9 million US dollar in the last 6 months of 2025. And then that ended up with the current loss of 6.8 million US dollar. So the grant money from the Solve FSHD and the X Prize is booked in the non-operating income, so it's not you can't see that in the revenue. Okay, so as I mentioned earlier, we have a good cash position even at this moment. However, we have a growth opportunity coming out from not just our lead program MDO-101, but also for MDO-201 and 103. So we wanted to fund the development of those the program. So we announced yesterday the new finance with EBO Fund. So the scheme of the finance is two parts. One is the straight bond of 500 million yen, and also 17 stock acquisition right, which consists of 192,000 unit or 19.2 million share equivalent stock acquisition right. So if the able exercise the warrant up to 500 million yen, it is allocated for repayment. But after that, that becomes the money we are able to use for development activity. So those amount of we're gonna raise will be used for additional funding for MD-101, as well as development of the 201, and also R&D costs, including MD-103 and the other, plus administrative costs. So this is another way of showing that we have raised a reasonable amount of money in the last finance announced last year. But on top of that, with the money we are raising, we will be able to fund development of MD-101, and also we can invest in development of 201, which has a great data, and we have a strong confidence that this will become the best-in-class solution for a DMD patient, as well as FSHD program. Okay, so nothing changed about the growth strategy, but I also wanted to highlight here that on top of MD-101, we have got a growth opportunity in 201 and also other program. So those are the next growth driver of Modality Therapeutics. And also, so this is another way of showing the pipeline status, but since we have a great progress in the DMD program, so this will become a much bigger opportunity for the company to help the patient. Okay, so maybe this is the last slide before the summary, but this is a not big. So this is showing where the Modality's core competence is. So we have a platform technology called CRISPR Gundam, and this is epigenetic technology to turning on or off the target gene specifically so that to treat the patient. But since we are focusing primarily on the payload, the content of the gene therapy, so we need to combine that with delivery technology. And we have established collaboration with JCR Pharma and GeneXcure. But as mentioned earlier, this time we are able to get access to the new state-of-the-art technology developed by Broad Institute. So our collaboration network is expanding to deliver better molecule for the patient. Okay, so lastly, this is the summary and the key takeaway of 2025 second quarter report is, first of all, we have a revised IND target for MD-101 to sometime in 2026. And the nextly, we have formed a license agreement with Broad Institute for mastotropic capsid to avoid the liver toxicity issue, which is great. So then the third point is MDO-201 and aim for a DMD gene therapy confirms strong efficacy beyond benchmark drug. That is the third point. And the next point is MDO-103 got for FSHD got 2 research grants one from XPRIZE and the other one from Solvay FSHD. So this make us to develop this molecule much faster with the collaboration throughout the collaboration with an academic group. And then lastly, we announced additional finance to invest in our new growth opportunities created by MDO-201 as well as MDO-103. So on top of MDO-101, we have a huge upside. We are seeing huge upside potential in our pipeline, and this fund, the amount of the money we are raising will support this development. Okay, so this is the end of the presentation, but since we got a multiple question from the investor and the shareholder prior to this meeting, so I'm gonna answer some of those questions. So first question is that, is there any impact from the recent toxicity issues of Elevidys? So I think I covered a part of the question in the presentation part, but I also wanted to mention that again, I wanted to mention that the hepatotoxicity is the growing attention in the safety viewpoint. So it is really important to make this less toxic to river. But luckily enough, we are able to get access to the state-of-the-art technology of masotropic capsid developed by Broad Institute. So this allows us to avoid those issues created by the natural occurring capsid, which is captured mostly by river. So that's the answer. The next point is then where in 2026 is Modalis expected to enter clinical trial. And also this is covered in the presentation part, and we are not able to disclose the detail of the timing. However, since we are in the review of the timeline, but we're gonna share the detailed timeline after we go through the development plan more precisely, and then once we get the raw granularity information, we're gonna share that to the public. Okay, then the third question is what are the terms of MyoAB license? So we can't disclose the detailed terms of the license agreement, but basically we're gonna pay to Broad Institute of royalty and also milestone payment as our broad development and the sales progresses. Okay, and then the next question is how much impact will US policy and top management change have? It's a difficult question to answer because there is so much uncertainty there. The bottom line is that whatever change is made in the US, the FDA or NIH or so the policy, we're gonna adapt. That's the short answer. And so as a part of the attempt, we started to explore our clinical trial, not just in United States, but in the other countries to prepare for any regulatory change or policy change in the United States. Okay, I think that is it. So thank you again very much for participating in the financial report and the business report of Modalis Therapeutics second quarter.